And there we go. Are there any questions? All right, if there's no questions, let's take a look at what we're going to be doing today. I need my glasses. I know we're doing chapter 3, and we should finish chapter 3. I'm hoping to get started on chapter 4, but I think we'll probably end early today. I doubt anyone will complain about that. And I don't even have chapter 4 starting today, but we're well along in chapter 3, so I think we'll finish it today. If you notice, there's no lab today. I will be on at 6:30 to 6:45 to answer questions. So if you have a question and you want to talk to me, make sure you log in between 6:30 and 6:45. I will be... Am I showing my screen? Yes? Okay. I will be there from 6:30 to 6:45. So if you have questions, make sure you're there from 6:30 to 6:45. If there are lengthy questions, I will be there longer, but if all the questions are answered at 6:45, I will log off. Any questions about that? So remember, in the lab, you do need to turn in the worksheet to Lab 4 by this Saturday. And let me see, I think there is a worksheet to Chapter 3, so that should be turned in by this Saturday as well. Any questions about any of that? If not, let's leave that.
Sir, you're actually not sharing your screen.
Oh, I thought somebody said I was. I need to get my microphone on. When I have, well, actually, I'm not sure. On the little one here, it's very hard to determine whether I'm recording it. And actually, last Tuesday, I did not record the lab. I think it was the lab. And I had to put on the recording from last term because I didn't have any recording in case people missed the lecture or the lab, whatever one it was. The good news is it covered the same material. All right, let me get this on. Now maybe I'll hear you guys better. So this was the slide that we had talked about last time, going over the gram stain procedure and showing you the different colors of the different cells at each step in the gram stain. You will need to know this. There will be questions, I think it's lab questions, on the gram stain, that after the gram iodine step, all the cells, gram negative and gram positive, are purple. And then after the alcohol wash, the gram positive cells are purple, the gram negative cells are clear. And then after the safranin step, the gram positive cells are purple and the gram negative cells are pink. Any question about any of that? All right, if no questions, let's move on. Let me erase this. Let's move on. We're now going to talk about the gram stain in intimate detail. So obviously you first make a bacterial smear on a glass slide and that's putting down the bacteria on the slide in a very thin layer. Ideally, you have the cells only one layer thick. You then air dry the slide and heat fix it so the bacteria cells are stuck to the slide. You then flood the slide with crystal violet. That's the primary stain for 60 seconds or 1 minute. And then after that time, you gently wash the cells with cold water. That's to remove the excess crystal violet. Crystal violet sticks to the peptidoglycan in the bacterial cell wall. It's considered the primary stain since all cells are colored by the crystal violet. So the color of the gram-positive cells is purple, the color of the gram-negative cells is purple. We then flood the slide with iodine. Iodine isn't really a stain, it is a mordant. Iodine can be used for a stain. If you're staining plant cells, for example, iodine stains, is it glucose or starch? I think it stains starch. But we're not using it as a stain, we're using it as a mordant. And a mordant is something that increases the affinity of the stain, the crystal violet in this case, for the specimen. What happens is the iodine complexes with the crystal violet and it makes a larger molecule. The color of the gram-positive cells is purple, and you could argue it's a more intense purple, and the color of the gram-negative cells is the same thing, purple or a more intense purple. Any question about that? After 60 seconds, you then wash the slide gently with cold water. You then flood the slide with acetone ethanol. Acetone and ethanol is the decolorizing agent, and this is the decolorizing agent that we use in the microbiology lab at Clark. Of course, we're doing it online, so I could really change it to anything I wanted to. But there are other decolorizing agents. In the last lab you had, it had a different decolorizing agent, which was just ethanol. And they treated it for a longer period of time than we treated. So when you're applying the decolorizing agent, if it's acetone and ethanol, you only apply it for about 5 seconds. For ethanol, you apply it for many seconds longer, and I don't remember if it was 30 seconds or a minute, something like that. And that's because ethanol is not as strong of a decolorizing agent as acetone and ethanol. You then wash the slide immediately with cold water. You need to do that immediately because you want to remove the trace amounts of the decolorizing agent from the cells. Even the trace amount of decolorizing agents left on the cells can pull out the crystal violet iodine from the gram-positive cells, and we don't want that to happen. So you wash the cells immediately with cold water. The acetone ethanol washes out the crystal violet iodine complex from the gram-negative cell walls, but not from the gram-positive cell walls, as long as you do not overwash. And let me state that this rule of thumb is only a rule of thumb. What you should do is hold up your slide and then apply the decolorizing agent and then watch the blue, the stain, or purple, run down. Once it stops running, you should stop applying the decolorizing agent. That's the best way to do that. because you can overwash, in which case you will remove crystal violet iodine from the gram-positive cells. And you can also underwash, where you will not remove the crystal violet and iodine from the gram-negative cells. So this is the trickiest step, and in one sense, ethanol is easier to apply because it's a longer period of time, and that means you got a few more seconds before you overwash or underwash. With only 5 seconds, I mean, you can overwash in certainly 10 seconds, and you can underwash in, I don't know, 2 seconds. So this step is tricky. All right. At this stage, this step is considered the differential step in the gram stain because the cells will look different. The gram-negative cells will appear colorless, and the gram-positive cells will look purple, assuming that you've done the gram stain correctly. All right, any questions about any of that? You then flood the slide with safranin. Safranin is the second stain that's used, so we call it the counter stain. And you apply safranin from 1 to 2 minutes. And then you wash the slide with cold water gently to remove the excess safranin. Safranin is a pink dye, and it does stain all the bacteria. It's a counter stain because it has a different color to the primary stain, the crystal violet. After applying safranin, did I mention, you then, after applying safranin, wash the slide gently with cold water to remove the excess safranin. After applying the safranin, if the gram stain has been done correctly, the color of the gram-positive cells will be purple. The color of the gram-negative cells will be pink. All right, any questions about that? Well, here's a question for you. I just stated that safranin stains all the bacteria. Why don't the gram-positive cells stain pink? Or you could say pink and purple together. Anyone have a guess? Come on, people, don't be shy.
Pink is close enough to purple that it won't overpower purple.
I think you got that right, and that is the purple is a stronger dye, and by our human eye, we only see the purple color. We do not see the pink color. But if you were to look at the cells under the electron microscope, you would see purple dye in one spot of the cell, and then pink dye in another spot. And of course, you look in other locations of the cell, you'll see more purple stain and more pink stain. So you do have the pink stain in there, but it's a less intense color than purple. And to our human eye, we only see the purple because it overpowers the pink. The pink does get in there. Okay? And that's not a trick question. I was just pointing out that why don't these cells look pink? Well, the pink does get in there, the safranin does get in there, but we don't see it with the human eye. You then blot the slide with bibulous paper. Let me see if I can show. Bibulous paper is a very thick paper. It's not quite as thick as cardboard, but it's actually thicker than regular paper. And you take the slide, pretend this is the slide. I don't have a slide here. I'm at home, as you can see. I should probably not show you that there. Let's go that way. And I don't know if you can see this. You then get the bibulus paper and then blot the slide. You don't wipe the bibulus paper on the slide because that'll wipe off the cells, but you blot it. So you fold it usually and then just press to get most of the water off the slide. And you get enough water off the slide so that you can then view the slide under the microscope, and you can view it even with the 100x immersion oil with oil. The traces of water are not enough to interfere with the oil when viewing the slide under the microscope. Any questions about that? So gram-positive cells are purple after the procedure. Gram-negative cells are pink after the procedure. Why? What's the basis of the differential staining? It has to do with the differences in the cell wall of prokaryotic cells. The gram-positive bacteria have a thick peptidoglycan layer, and the crystal violet iodine gets trapped in that thick peptidoglycan layer and doesn't wash out. The gram-negative bacteria have a thin peptidoglycan layer in their cell walls, and it also has a lipopolysaccharide layer as part of the cell wall. We call that the outer membrane of the cell wall. And the important part is the thinner peptidoglycan does not trap the crystal violet iodine in the cell wall, and it washes out in the decolorizing step. So let me see if I can blow this up. What the heck? Oh, here we go. The gram-positive cell wall is here, and it shows you a very thick cell wall that's purple right there. That's the cell membrane that's yellow. Cell membrane right there, a lipid bilayer. And the main molecule in the cell membrane, of course, is phospholipids. We talked about that before. Peptidoglycan is the main molecule in the cell wall. And the peptidoglycan layer is very thick. Now, this artist's rendition is actually only showing you 3 layers of peptidoglycan. In reality, it's much more than that. It's probably several hundred layers of peptidoglycan. Okay? Any question about that? And then with the gram-negative cells, they have a thin layer of peptidoglycan and a lipid outer membrane. And so we have right there the cell membrane, which is mostly phospholipids. And then on the outer part of the cell wall, we also have lipids. And let me see if I can point this out. The lipids on the lower bilayer is familiar to you. It's actually phospholipids. In the outer bilayer of the outer membrane of the cell wall, there are phospholipids, but there's also lipopolysaccharides, and that's this blue molecule here with the hair. And the important part is lipid A, which is the big blob right there. That's a lipid portion. We'll talk a little bit more about that later. The important part is on the gram-negative cells, they have a thin peptidoglycan layer. And the artist is only showing you 1 layer of peptidoglycan. And that's not correct. Come on, mouse. There's more than one layer of peptidoglycan, but the layers are much less than in the gram-positive cells. Okay? And I'm not sure how many layers of peptidoglycan is, but it's more than one. Let's say something between 5 and 30. It may even be 100. I'd have to look it up exactly. The point is, it's much thinner in the gram-negative cells than the gram-positive cells. Any question about that? And the crystal violet on the gram-positive cells gets stuck in this thick peptidoglycan layer, and it will wash out of the thinner peptidoglycan layer. I've already mentioned this, the iodine complexes with the crystal violet making a larger molecule. In the gram-negative cells, the crystal violet iodine is washed out because the peptidoglycan layer is thin, and the lipopolysaccharide layer is actually disrupted by the alcohol wash. And then the cells are colorless. The gram-positive cells, the crystal violet iodine is not washed out because it's a thicker peptidoglycan layer, and the cells remain purple. So here's a gram stain, and you can see gram-positive rods, a gram-positive coccus, a gram-negative spirilla, and a gram-negative rod. Any questions about that? The gram stain has some technical problems, like what do you do when you see a slide looking like this? What that is and that there is actually a crystal violet crystal, meaning when we have a crystal violet solution, unless it was freshly made, the crystal violet crystallizes over time. And especially if you're using from the bottom of the crystal violet, you'll get these crystals on your slide. And this we consider a bad region of the stain, that too, and you don't want to determine your gram stain around these bad regions of the slide. So these are bad regions of the slide, and then you determine the gram stain using the good regions of the slide. I've actually seen it where somebody had gram-negative cells, and they had crystals of crystal violet on the slide, and the crystal violet from the crystal went out and stained purple the gram-negative cells immediately around this crystal. But if you look at the good regions of the slide, the cells were staining pink from the safranin. And so the point is, don't do your gram stain in bad regions of the slide. The gram stain has a number of problems. The most common problem is that students do not always use young growing bacteria. To get the gram stain to work consistently, you have to have young growing bacteria. What that means is the cells should be growing for 2 days or less. And if it's over 2 days, then they're not young growing bacteria, and the gram stain may not work correctly. The gram stain is not universally applicable. Some bacteria stain poorly or not at all. It's mainly the mycobacteria, and that's because the gram stain is a water-soluble stains, and the mycobacteria have a lot of lipid, mycolic acid, in their cell walls, and so the cells do not readily take up the stains of the Gram stain. I have actually used 2 specimens of mycobacteria, and as long as you did the Gram stain correctly, stain it on freshly growing cells, and make sure you leave the crystal violet on for at least 1 minute, I did get the mycobacteria to stain correctly. Okay, meaning they did take up the Gram stain, the crystal violet. Mycobacteria, if you don't know, are gram-positive cells. But sometimes they will be lighter staining than other cells, and it's just because the cell wall has a bunch of lipid in it, and it doesn't like taking in water-soluble stain. The Gram stain results are not always as clear-cut as theory suggests, and this will become evident during your unknown project, and you're lucky you're not going to be in the lab, because students in doing the Gram stain, some of them didn't get it to work very well, and they were literally pulling out their hair because, well, the Gram stain is tricky. Okay, and it's mainly the decolorizing step that's tricky, and that is you have to decolorize for the right amount of time. You can't over decolorize, you can't under decolorize. And the other thing is, make sure you're staining freshly grown cells. The structure of the gram-positive and gram-negative cell walls not only affects the staining as applying to the Gram stain, it also is related to how they respond to antibiotics. For example, gram-positive bacteria are easily killed by penicillin G, as well as by cephalosporins and many detergents. And that's because the gram-positive bacteria do not have an outer membrane, and so the penicillin G, the cephalosporin, and the detergent can get at the cells and then kill them. The gram-negative bacteria are, on the other hand, more resistant to these antimicrobials, like penicillin G and cephalosporin and some detergents, and that's because they cannot penetrate the lipopolysaccharide layer that we call the outer membrane, of the gram-negative cell. Penicillin G is not effective on gram-negative cells because this outer membrane prevents the penicillin G from getting into the cells and then doing harm to the cells. Any question about that? Now, we can use other antibiotics to kill gram-negative cells. For example, ampicillin, which was an antibiotic made, I think, in the early 1960s. It's one of the early members of the penicillin family. Ampicillin does work on gram-negative bacteria. It also works on gram-positive bacteria. And there are other members of the penicillin family. I'm trying to remember. Another one is carboxycillin. It works on both gram-negatives and gram-positives. All right, any question about other differences between the gram-positive and the gram-negative cells? All right, let's briefly talk about special stains. You're not going to be examined on special stains, at least not in the lecture. You could be in the lab because our last lab discussed special stains. But I do want to talk just briefly about the capsule stain because, well, it's a stain used to stain for a capsule, and some of you may need to use this stain later in the term for your lab unknowns project. I'll be giving you a bacteria, and if you think your unknown bacteria has a capsule, you may wish to do the capsule stain. If you do, the capsule stain stains the background of the slide, right here, and it stains the cells, but it does not stain the capsule. Let me see if I can, well, let's go to the next slide. India ink is another capsule stain. It's actually a negative stain. It lightly stains the cells and darkly stains the background, but the capsule does not stain with India ink. And so if you see this on the capsule stain, then you know your cells have a capsule. All right, any question about capsule stains? If not, that's it for chapter 3. I thought I already saved that. Let's move on to chapter 4 and we'll just get started with it. We're only 34 minutes into this lecture. All right, chapter 4, as is the case with me, I try to give you a first slide telling you the name of the chapter, chapter 4, the functional anatomy of prokaryotic and eukaryotic cells, giving you the major goals of this chapter and a rough outline. Of course, you need to know the terms. What's a coccus? What's a bacillus? What's a sporilla? What's a strepto, like a streptococci? What's a staphylo, like staphylococci? What's a diplo, like diplococci? What's a tetrad, like a tetrad of cocci? What's sarxinia? What's a vibrio? What's a spirochete? What's the glycocalyx? What's a flagella? What are axiofilaments? What are fimbriae? What are pili? What are peptidoglycan? And we kind of already talked about that one. What is an endotoxin? And what is selective permeability? What is diffusion? What is facilitated diffusion? What is osmosis? What is active transport? What is tonicity? What is plasmid? What is inclusions? What are endospores? And what is the endosymbiotic theory? Now, let me state that we will be talking about peptidoglycan in this lesson and actually the previous lesson. So you know that there will be a quiz question on that and that there might be even a final exam on that. And this will be the first time we're talking about endotoxin, but we'll be talking about endotoxins in 3 places in this lab. So once again, these 2 are major topics that we will hit time and time again in this class. And I guess here's another one, an endospore. We will talk about that at least in 2 times. I'm not sure how many, maybe 3 times. And so that these are ones that you do need to know, and we'll talk about them time and time again. All right, you do need to know how gram-positive and gram-negative cell walls differ, and we'll go into that in a little more detail. You already know that they differ in their thick versus thin peptidoglycan layer. There are a few other differences too. And then lastly, be able to describe the importance of taxis in bacteria. Any question about what we're doing? All right, so we're going to talk about prokaryotes and eukaryotes. And prokaryote is a Greek word meaning pre-nucleus. Pro means pre, and karyotes, well, literally it means kernel, but we're taking it to mean nucleus. And prokaryotes don't have a nucleus, so we call it a pre-nucleus. Eukaryotes, eu means true, and karyotes, we're using that word to mean nucleus. The prokaryotes generally have one circular chromosome, and the chromosome is not in a nucleus. What do we mean by a circular chromosome? Meaning the chromosome is in a circle, and if I start right here, I end up right here, meaning the start and the ending point is in the same place. Any question about that? Eukaryotes have generally several chromosomes. Humans have 46. You don't really need to know that, but we do have 46. And the chromosomes are linear. So if we look at the X chromosome, it is a starting place, and then down here would be the ending place. And the starting place is different from the ending place. And that's what we mean by a linear chromosome. The chromosomes of eukaryotes are in a nucleus, and if you don't know, the nucleus is a membrane-bound structure that contains the chromosomes of eukaryotes. In prokaryotes, they do have DNA, and it is associated with proteins, but it is not associated with histones. Histones are a protein that we find in the DNA of eukaryotes. And actually, when we look at chromosomes of eukaryotes, we find that they're made up of both DNA and proteins, and the major protein is histone. Any question about that? If you don't know, the DNA of eukaryotes wraps around the histone protein. And that protein is not found in prokaryotes. Prokaryotes do not have any organelles. By organelle, I mean a membrane-bound organelle. And eukaryotes have many organelles, and I mean membrane-bound organelles, like the Golgi apparatus and the vesicles and the endoplasmic reticulum, the nucleus, the mitochondria, okay? Just to name a few of the organelles. Bacteria have peptidoglycan in their cell walls, and you probably should know that peptidoglycan is only found in bacteria. The archaebacteria do not have peptidoglycan. And the archaebacteria generally differ from the bacteria in regard to peptidoglycan. The eukaryotes have no peptidoglycan in their cell walls, and of course animal cells do not have cell walls, so we don't have peptidoglycan either. Prokaryotes, when their cells divide, they mostly divide by binary fission. Binary fission is where one cell literally splits into two. Eukaryotes have cellular division by mitosis, and this is a topic hopefully you learned in Biology 160, because we're not really going to talk about it. But they do differ on how prokaryotes and eukaryotes undergo cellular division. All right, any question about this overview of prokaryotes and eukaryotes? So this slide is showing you a summary of what we're going to be doing. We're first going to talk about prokaryotes. We'll talk about the cell shape and the cellular growth arrangement. We'll then move into the structures of cells, and I call those the cell structures. And then lastly, we'll talk about the movement of material across membranes, particularly across the cell membrane. And we'll then talk about eukaryotes, and we'll talk about the cell structures in eukaryotes. And a lot of these cell structures are organelles, but the ribosomes would be a cell structure that's not a membrane-bound organelle. And then lastly, we'll talk about the endosymbiotic theory. All right, so when we're talking about prokaryotic cells, they have a particular size and shape. The average size ranges from 1 to 10 micrometer, and really the average is most cells range in size between 1 and 2 or 3 micrometers. Okay? 10 micrometers is on the large size of a prokaryotic cell, and there are some that get less than 1 micrometer. I think the smallest prokaryotic cell is around 0.4 micrometers. And like I said, different people have different average sizes, so I'm not going to quiz you on what's the average size of a prokaryotic cell, but you should know that the average size is smaller than a eukaryotic cell. Prokaryotic cells have 3 basic shapes. There are coccus, which are round ball-shaped cells, right there. There are bacillus, which are rods, and there are spirals, spirilla shown here. Those are the 3 basic prokaryotic shapes. Most of the cells have these shapes or some derivative of these shapes. And I'll talk a little bit more about that in just a bit. This slide is showing you the different types of spirals, and you could call a vibrio a half spiral, because here's the spirillum, and a vibrio is sort of like this portion of it. So you'd say about a half or maybe a third of a spiral. You could also call it a bent rod. Either one, it is a shape based upon the 3 basic shapes. It's either a bacillus or a spiral. And here we have a spirillum. The spirillums are rigid. They tend to spiral in a corkscrew type shape. You can also have a spirochete, which has many bends in it of the spirillum, and they are more flexible, bendable than the spirillum. Any question about the different types of spirals? You can have prokaryotic cells which are of different shapes, and we call them unusual shapes, like the Stella bacteria. The bacteria actually have a star shape, and here's an actual electron microscopic image of Stella. And then the Haloarcula is an unusual shape, and this Archaea bacteria actually has a square type shape or slightly rectangular shape. Most cells are not the shape. Most bacteria species are monomorphic. That means they have one shape for all the cells in the species. Like the Haloarcula, all the cells are squarish. But some cells, really just a few, are pleomorphic. And the cells take on many different shapes. Pleomorphic means many shapes, and or many morphologies, shapes. And here we're looking at a pleomorphic cells. Let's see if I can remember this family of bacteria that tends to be pleomorphic. Mycoplasma. The family of bacteria that tend to be pleomorphic is the Mycoplasma. And they don't have a cell wall, and that's why they take on many different shapes. Let's talk about the cellular growth arrangement. The cellular arrangement, or I like to say the cellular growth arrangement, is how the cells grow and then stick together. For cocci, you can have single cocci, where there's only one cocci. Or you can have diplococci, where two cocci stick together. And here we see a diplococci. And I don't know why that's not a better picture, but it isn't. And if this keeps growing like that, we can have streptococci, where the cocci line up in a row. You can also have cocci in a tetrad, 4 cells stuck together. And what happens is the cells divide on this plane, and then on that plane, and then they divide, let's see, they divide on this plane, making that one, and then both of them divide on this plane, making these two. And you can have Sarcenia, which are 8 cells stuck together. 1, 2, 3, 4, 5, 6, 7, 8. And you can't really see that in these pictures, but trust me, this is a tetrad. Actually, you can see the tetrad right there. And if anyone wants to try to count that, that's an 8. There's also cocci that can grow in a staphylococci, and the cells just grow together like a cluster of grape cells. And if you look at this looks like a cluster of grapes. And staphylococci actually means cocci growing in a cluster like grapes. Okay? That actually is what that means. The bacilli also have cellular growth arrangements, or called cell arrangements. And the first cell arrangement you can have is a single bacillus, where all you have is one bacillus, nothing stuck to it. You can also have diplobacilli, where 2 bacilli are stuck together. And I don't really see that in this picture here. Maybe that one there. It's a pair of bacilli. You can have streptobacilli, and this is a chain of bacilli, and they grow in a sausage link. In the old days, you could buy sausage links where they were actually hooked together like that, and that's streptobacilli. And then lastly, you can have the bacilli stuck together along their, I don't know, short side. And they're stuck on this axis here. And this is called a palisade. If you don't know what a palisade fence is, this is the fence that Mark Twain and Huckleberry Finn were painting in, what the heck is that name? Was it Huckleberry Finn? Or Mark Twain? Whatever. A palisade fence, you take this shape and then you rotate it 90 degrees, and then this part of the fence is put into the ground, and then the board is put up to make the fence. And they're not very common today, but in Mark Twain's day they were very common. Any question about any of that? Those are the different cellular growth arrangements that bacilli can have. And you'll note that bacilli have fewer growth arrangements than the cocci. Now there's something I want to talk about here, and that is a coccobacillus. This is a shape in between a coccus and a bacillus. So if you have a round bacillus, which rounded on the ends, and you get it smaller and smaller, you end up with what we call a coccobacillus. And there are a few species of bacteria which are coccobacillus. You might even have one for your unknown bacteria. I'm not sure why this is put in with the bacilli, because this is a coccobacillus, not a bacillus. And you could argue it's as much of a coccus as it is a bacillus. All right, any question about any of that? Now, let me warn you that when you see the term bacillus, it has two meanings. If it is a capital B, it means it is the family of Bacillus. So there are different genera or species within this family. Don't get that confused with the shape, and we will always use a lowercase b to mean a rod-shaped cell. Okay? And the same thing with streptococci. Streptococci is a growth arrangement of cocci. If we ever spell it out with a capital S, and the name is Streptococcus, this is a family of bacteria. Actually, strep throat is an example of Streptococcus. And I'm trying to remember a bacillus. I don't know if you know Bacillus megaterium or Bacillus, I'm drawing a blank here. Bacillus subtilis. Those are two species in the Bacillus family. And like I said, look for the capitalization. If it's capital, that means we mean a family. And if it's not capital, then we mean a shape of the cell. And of course, Streptococcus is the family, and we will always give it the name Streptococci for the cellular growth arrangement. And it should be a lowercase too. All right, any question about any of that? If not, let's look at some structures of a typical prokaryotic cell. This is an artist's rendition of a bacteria, and here we have an actual electron microscopic image of a bacteria cell. There are 4 regions of the bacteria that you find in all bacteria. All bacteria have cytoplasm, all bacteria have ribosomes, all bacteria have a plasma membrane, and all bacteria have a nucleoid region containing the DNA. Now you should know that the DNA is dissolved in the cytoplasm, and it moves around in the cytoplasm. It just sort of floats around. Wherever the DNA is floating, that's the region we call the nucleoid. And then the rest of the region is non-nucleoid. But there is no boundary between the nucleoid region and the rest of the cytoplasm. And this nucleoid region could move, assuming the DNA floats over here, okay? And that's just the way prokaryotes are. Why do we call the nucleoid region a region of the cell? It's just an old term that microbiologists use. When they first started staining cells, they noticed that the nucleoid region stained differently than the rest of the cytoplasm. And the reason it stains differently is because DNA stains differently. It's a real large molecule, and so it takes up different characteristics of the dye. Usually it stains darker if you're only using one dye. This one it looks like we're using two dyes, and the nucleoid region is taking on a bluish color, and the cytoplasm is taking on a pinkish or reddish color. So remember on prokaryotes that the DNA is the nucleoid region is wherever the DNA is floating in the cytoplasm. There is no boundary between the nucleoid region and the cytoplasm. In eukaryotes, there is a boundary, and that we call the nuclear membrane. Okay? Any question about any of that? All right, the plasma membrane is found on all prokaryotes. It's also found in eukaryotes. Ribosome, found in prokaryotes, all prokaryotes, and in all eukaryotes. And the cytoplasm, also found in all prokaryotes and in all eukaryotes. So generally speaking, these three, and then you can say DNA is a fourth, are all found in cells. The cell wall is found on most prokaryotes, and it's outside of the cell membrane. Let's see if I can blow this up. So we have the plasma membrane, which is yellow here, and it's immediately after the cytoplasm, which is pink. And I don't know what that pink line is, because the cytoplasm doesn't have a line. That's just the artist's rendition, and you wouldn't see that on an electron microscopic image. But you can see the cell membrane, or the plasma membrane, on the electron microscopic image. And here it's right there, but as you notice, the cell membrane is right next to the cell wall, and so you cannot generally distinguish them. Oops, sorry. To distinguish them, you can put the cell in, actually we might be able to see, now that I've blown it up. Yeah, see right here? We can see the plasma membrane. This line right here is the plasma membrane. And then that's the cell wall. It's immediately outside of the cell membrane. Shrink that. And then outside the cell wall is the capsule. Remember that not all cells have a capsule. Only some of them do, and if we can see it under the light microscope, we call it a capsule. It is a structure that has other molecules in it besides water. If you can't see it under the light microscope, but there is a glycocalyx outside the cell wall, then we call it a slime layer. And to see the slime layer, you'd have to look at it under the electron microscope. The light microscope can't see it because the slime layer is mostly water. The capsule and the slime layer are secreted external to the cell, and they're secreted by the cell. So the last part of the cell is the cell wall. Everything external to the cell wall, we say, is outside the cell. And so the capsule is outside the cell. You can have other structures in the cell. Fimbriae are hair-like structures on prokaryotic cells, and they don't move much. The cell can retract them. They're usually used for attaching to the cell to things. Flagella is much longer than a fimbriae, and it's made of flagellar proteins. And this is a whip-like structure, and actually we see several of them here, which the cell uses for swimming through a liquid media. So the flagella give the cell movement. A plasmid is a small molecule of DNA that we can find in prokaryotic cells, and it's separate from the chromosome. And lastly, a pilus, a sex pilus, we'll talk more about that later. That's a structure on some cells which is used for moving plasmids from this cell to another cell. We'll talk more about that later. And then inclusion bodies are things that the cell has brought into the cell or made in the cell, and they accumulate in one region of the cell, and they get to be so many of them that they actually precipitate out of solution, meaning initially they're in solution, the cytoplasm, they're dissolved in the cytoplasm, but occasionally cells accumulate so much of a certain inclusion that the material precipitates out of solution, and then we see a dot or a glob in the cell. And does anyone see an inclusion here? It doesn't have an inclusion over here, but that could be an inclusion, and that could be an inclusion too. All right, any question about any of this? If not, I think we will stop here, and I will see you at 6:30 for the lab. And there isn't a lab. I'm going to be there from 6:30 to 6:45 to answer questions. And if there aren't other questions at 6:45, I'll log off. Of course, I will stay there to answer all the questions, but I'm there to answer your questions. All right, any questions?
It's just the same lab from Tuesday, right?
Let me turn that up. I didn't hear that.
There's only one lab this week, right?
Yes, lab 4 is the
Okay.
only lab this week.
Got it. Thank you.
Okay? All right. Well, I'll see you at 6:30. If you have a question, you don't need to show up if you don't have a question.